Yo no sé de dónde salís.
Conectados online. Mi nombre es Jesús Santamaría y me han pedido que presente a nuestro invitado esta tarde, cosa que hago con mucho gusto, en una de las sesiones de los Thursday NanoSpin-off Talks, que como sabéis, lo que pretende es, por un lado, traer experiencias de spin-off desde el ámbito universitario, desde el ámbito de investigación, con el objeto de que las conozcamos y además de que inspiren a la gente, sobre todo a la gente más joven que tenemos en la audiencia, para que algún día se pueda lanzar al ruedo. Y eso es lo que ha hecho nuestro invitado esta tarde. Nuestro invitado esta tarde es el doctor Víctor Francis, que sin haberle conocido prácticamente nada ya le admiro, ¿no? Porque es una persona que es doctorado en biología en la Universidad de Barcelona, su doctorado en enfermedades metabólicas, y de repente, en un momento determinado de su vida, decide darle el salto a una spin-off. Una spin-off que tiene, ahora nos lo contará, tiene un año de vida, un año de poquitos días, y no ha ido a esa spin-off en su faceta de científico, sino como business developer. O sea que, si miráis en la página web de la empresa que se llama DIVERSA Tecnología, está encargado de establecer acuerdos colaborativos, costes, calidad de los productos. Tiene un montón de responsabilidades que no tienen que ver directamente tanto con hacer más investigación. Esto la hizo, es un investigador que se ha pasado, algunos pensaréis que al lado oscuro y otros pensaremos que al lado brillante, ¿no? Es la dicotomía de, bueno, doy un salto en el vacío y vamos a ver qué es lo que pasa. Entonces, hoy nos presentan, desde el punto de vista de un científico, una empresa que está fundada por científicos, y si vais a la página web veréis que hay dos doctoras en biología molecular, creo, y un médico, que son los que han lanzado la empresa. Es una empresa joven, pero ya tiene como 10 personas contratadas y está en la fase inicial. O sea que tenemos la suerte de ver un proyecto en la etapa más emergente justo cuando están enfrentándose a los problemas iniciales, no tan iniciales, después de un año, y todo eso nos lo va a contar, aparte de contarnos de qué trata, y podremos entrar en el debate tanto, espero, desde el punto de vista científico como desde el punto de vista económico, de cómo se hace esto y qué es lo que pasa. Solo agradecerte una vez más que has venido aquí, invitado por la Cátedra Sanca de Nanotecnología, y que te podamos conocer más y ojalá hacer algo juntos también. Así que bienvenido, Víctor, y the floor is yours.
Thank you very much. Inglés, ¿verdad?
Ay, yo he hecho la...
No pasa nada.
¿La hago en inglés o te la repito en
No, no
inglés?
te preocupes.
Vale.
No, sí,
So sorry.
Okay. Well, good afternoon, and thank you very much, Jesus, for the kind words. My name is Victor Francis. I'm Business Development Director at DIVERSA Technologies. We received the invitation from Marta Jimenez, and we're very thankful to the Cátedra SAMCA de Nanotecnología for the invitation. It's really an honor to be presenting what we do at DIVERSA and our technology, and also thanks to the audience, to the nanotechnology master students and the INMA researchers, and all the audience at home as well. So we started as a spin-off from the Health and Research Institute in Santiago de Compostela in Galicia. I started 1.5 years ago with the company, but the research has been going on for more than 10 years. Maria de la Fuente is the PI that started the lab and also ventured into incorporating the company, DIVERSA. The reason we started DIVERSA is because 150,000 people die every day of diseases, and many of them have no treatments at all. Of the 5,000 rare diseases that are known, only 7% have a cure at all. And this is not because there are no treatments or no molecules discovered. It's just that these molecules cannot make it to the clinic. So there's a gap, there's like a valley of death between the molecules that are discovered and taking them to the clinic and to the patients. Actually, it's 3% of the new molecules discovered these are the only ones that will make it to the clinic, and the rest, 97%, will never make it. So we started with the idea of taking pharmaceutical nanotechnology to the clinic so that we can improve patients' lives and also help get new therapies to the clinic faster. Our technology is based on lipid nanoemulsions, and we'll talk a bit more about them afterwards. They're biocompatible and biodegradable, and they can be used for new drug developments or new therapy developments in the pharmaceutical, cosmetic, and nutraceutical industries. We have designed them with translatability and simplicity in mind. This means that compared to pre-existing technologies like lipid nanoparticles, for example, you've always heard about them. You probably have them in your body if you got the COVID vaccines based on mRNA from Pfizer, BioNTech, and Moderna. Our nanosystems are simpler in the composition, which means a more cost-effective manufacturing and also less regulatory hassle, because the two main components are just 2 compared to 5 or more with lipid nanoparticles, for example. They are safe by design, they're versatile, they're reproducible, and they're biocompatible. This is how we've designed them to be from scratch. The patent is the cornerstone of the project. Usually for any spin-off not coming from an academic institution, there's a discovery, and then you need to decide, am I gonna publish this or do I keep it secured until I've published the patent? So we decided to go for the patent because we wanted to constitute the company. It is approved already in the European Union. It's in national phases, and it's pending for approval in other main markets like the US, Japan, Korea, Russia, and Brazil. We were lucky enough to have lots of support, mostly financial support, like any startup. So we got grants from the Xunta de Galicia, from CaixaBank, and also from the Instituto de Salud Carlos III. But we also had, and it's as important to get financial support, we had support from more institutional partners. And a special mention to BIOGA, which is the Galician Biotech Association, and because they help all of the startups in Galicia grow faster. And it's really, I think it's a smart move if you're a bio entrepreneur to go and join your local, the Aragon Biotech Association. The company incorporated in January 17th, 2022. So we are like 1 year and 9 days old. We're very young indeed. After doing all the market research and all the filing the patent, we were able to incorporate. We actually count with institutional partners like the Foundation of the Health and Research Institute of Santiago and the SERGAS, which is the Servicio Galego de Saúde. So it's the health branch of the Xunta de Galicia. The co-founders, or the company, is a mix of biologists, pharmacists, medical doctors. The co-founders, we have Maria La Fuente, who is the CEO and CSO, and she has more than 20 years experience in nanotechnology. Some of the researchers here at INMA know her from the research lab. Abby Vázquez is our CEO. She did the PhD with Maria, and she's co-inventor of the patent. And of course, we have Rafael López, who's the medical advisor, and he is the head of the oncology department at the University Hospital in Santiago. And he keeps us connected to the research and the clinic over there. Then we have a talented team as well. We are actually 7 employees. We have the 1 patent, and we've started already commercial activities in Europe and India and Japan. On top of that, we have an advisory board, which is a mix of business and scientific advisors. And this is also a very important figure for a new company, for a startup, because you need to rely on people that have know-how on things that you have no idea and you didn't even know that you had to know before you started the company. So they're a really good asset of the company in terms of reaching out to them when we have any kind of problem or that we don't know how to solve. So with all this hard work and all this help, we constituted DIVERSA. And again, just to reiterate, the idea of starting the company was to solve this problem of only 3% of the molecules discovered in research labs around the world, making it to the clinic and to the patients. The reason they don't get to the clinic is because they're not soluble, or they get degraded once they're in the body, or they're not able to enter the inside of the cells, which is where 3 out of 4 new molecules have their targets. With our technology, we want to give new opportunities to some of these molecules to make it to the is based on lipid nanoemulsions, and they're self-forming and in the nanometric scale. This is the PDF, so the video probably won't work, but it was just showing how easy it is to reconstitute the mix of lipids, and with a method that's called ethanol injection, reconstituting lipids with ethanol and then water or the buffer that you're going to work with, and they're already formed. There's no need for special microfluidic systems or special equipment. They are self-forming nanoemulsions. They are in the nanometric scale, usually between 100 to 200 nanometers, but we can regulate the ratio of the components in order to change the diameter of these nanoemulsions. Then the other interesting characteristic is the electrical charge. And here you can see the zeta potential. It's a representation of the electrostatical charge of these molecules. It's very close to neutral charge. And this tells us that the molecules are not going to be sticking to other molecules rather than just the cargo. And this is a difference with other technologies that use cationic lipids, for example, to bind the cargos. So one advantage that we have is because we do not use the cationic lipids, we have less toxicity because of the lack of these off-target effects. We're also very stable. So we've checked the stability of these nanoemulsions in different conditions. The first graph is storage in different humidity and temperatures, and it's in ICH, long-term stability study. So what you see here as 6 months, it's actually 2 years. And this mostly we've done for one of our products, which is an off-the-shelf product that I will talk about later. We've also done stabilities in shorter times, like in PBS and in different cell culture media, buffers, sera. The important point here is that we're stable with FBS. And this is, again, a difference with some of the pre-existing technologies, because FBS, which is very rich in many proteins and molecules, will mask these electrostatical interactions. Because we do not rely on this to bind the cargo molecule, we can use FBS, or the researcher can use FBS when they're using our nanoemulsions. We also check stability in different biological relevant fluids, like human plasma, tear fluid, synovial fluid, and gastric and intestinal. And this gives us an idea of the stability of our nanosystems in different administration routes. Of course, the nanoemulsions have been designed to be used both in vitro, in vivo studies, and even to take them to humans. So we wanted to check biocompatibility. We did this in different cell lines. Here we show fibroblast and hepatic cells HepG2. And you can see that with increasing concentrations of the nanosystems, we didn't see a decrease in the viability of these cells. The next graph, top right, is the experiments in mice. We use very high doses of the nanosystems, which would never be used in the clinic, in two different ways. One was a one-time injection of 30 milligrams per kilo. The other one was 3 injections at days 2, 4, and 6 of 60 milligrams per kilo. In both cases, the animals, the weight didn't change, the behavior didn't change, and of course, none of the animals in the experiment died. So telling us that these nanosystems are safe to be used with mice. And the graph below is with zebrafish embryos. In this case, we used, again, increasing concentrations, and we didn't see a difference between control and very high concentrations in these animals. All these experiments telling us that the nanosystems are safe to be used in vivo models. Another interesting thing is the cell internalization. So we checked how fast the nanosystems would enter the cells, because of course we want to use them as carriers for molecules that cannot enter the inside of the cell. And you can see, well, these experiments, we did them with the nanosystems that were empty, but were labeled with a green fluorochrome. You can see the image of the fluorescence microscopy underneath, and the top left graph, you can see after 60 minutes, 100% of the cells had the nanosystems inside. This is a very fast internalization. We tested a battery of cell lines, even primary cultures of astrocytes, and you can see that for all of them, in this case, it's 2 hours, all of the cells had the nanosystems inside. So it's a very good system to take molecules that otherwise would not get inside the cells in a pretty fast way. Then we went into studying the biodistribution of these nanoemulsions when they were injected in animals. So in this case, we tried 3 different administration routes with radio-labeled nanosystems. We used zirconium-89 as the labeling agent, and we did intravenous, subcutaneous, and intraarticular injections. And in all of the cases, we had accumulations for at least 48 hours, depending on the experiment. We also went a bit more in detail to study the distribution in different tissues. In this case, we used gallium-68 that was labeling the nanosystems, and we did PET scan in mice. The yellow block here in the image, it's the liver of these mice. And you can see in the graph that the liver is the tissue that accumulates most of these nanosystems. It is normal or usual for lipid-based drug delivery systems to accumulate mostly in the liver. But we were also able to observe significant amounts in the blood, kidneys, spleen, and lungs. And the graph on the right is to tell or illustrate that by changing, for example, the administration route. Here we're comparing intraperitoneal to intravenous injections. And you can see the graph in the bar in white, the liver at 4 hours. It's very different, the accumulation, based on the administration route. We also have other strategies to change this accumulation in liver. For example, changing the dose or decorating the nanosystems with targeting peptide or antibody, and also pre-treating the patient or the animal with empty nanosystems before administering the nanosystem with the therapeutic molecule. We have a very versatile technology when it comes to binding different types of molecules. So we can bind small molecules, peptides, proteins, and different types of nucleic acids. all of them with very high association efficiencies. This is a crude representation of the nanosystems. The inside is hydrophobic, and the outside is a lipidic monolayer, which is formed mostly by sphingolipids, sphingomyelin. So the outside has some hydrophilic groups to bind hydrophilic molecules. Here are some examples of small drugs that we encapsulated with our nanosystems. We check different molecular weights, different hydrophobicity, that's the second column, the log P, and all of them were encapsulated with very high efficiencies. We can also encapsulate different types of peptides. We've tried peptides of different lengths and different isoelectric points. The image on the left illustrates how a free peptide that's labeled with green fluorochrome, when it's incubated in the cells and it's free, it doesn't enter the cell. So the blue staining is DAPI for the nuclei, the red staining is rhodamine for the cytoskeleton, and then the peptide is labeled in green. When you incubate for 2 hours and you wash the cells, you can see that there's no peptide whatsoever. When the peptide is associated to DIVERSA nanosystems, it's able to enter the cell and it's colocalizing with rhodamine, so it's in the cytoplasm. With proteins, we have similar results. So we've tried different molecular weights from very small proteins from 17 kilodaltons up to 540. And we can also bind different proteins with different isoelectric points because we use two binding strategies. One is using covalent binding, so it's good for any kind of protein. The other one is only able to bind anionic proteins because it's using ionizable lipids. So in this case, we are using electrostatic charges, but we have both strategies. On the graph on the right, we're just seeing the stability of these nanosystems at 0 and 24 hours, just to check that the size doesn't change and the nanoparticles are stable. This is a very interesting result comparing the biocompatibility of our nanosystems to a commercially available competitor. So in this case, what we did is we incubated cells with free beta-gal, this 540 kilodalton protein. On the left, you can see we incubate with a free protein. You wash the cells and it barely enters the cells. When beta-gal is associated to DIVERSA reagents, it enters the cells and it's active because the blue coloring comes from a substrate of beta-gal that gets transformed and gives the coloring. With a competitor of ours that in this case it's based on liposomes, you can see that beta-gal also enters the cells, but the cells look not very healthy and there's a lot of cellular debris. So this is just after 2 hours incubation in vitro. You can imagine if you were using this for an in vivo model. So we really think we have an advantage in terms of less toxicity and more biocompatibility. We can also use our nanosystems for gene therapies, and we've tried with different types of oligonucleotides, siRNAs, microRNAs, aptamers, mRNAs, plasmid, and complementary DNAs, all of them with very high association efficiencies. When you see here the length size or the range of lengths that we've tried, this doesn't mean that it's the edge of what the technology can manage. It's just what we've tried so far. Some examples of gene therapies that we've tried is plasmid DNA with some breast cancer cells. We usually characterize the nanosystems. We see that they're in the usual 100 nanometer scale. We do association efficiencies running an agarogel, and you can see that the free plasmid is able to migrate, and the plasmid that's associated to the nanosystems doesn't migrate because it's all bound. And then we looked at the therapeutic effect. In this case, it was anti-tumoral plasmid DNA. So the scrambled plasmid DNA was used as a control versus the plasmid DNA in the light blue with anti-tumoral effect. And we compared with increasing concentrations of these plasmids, how the cell growth was diminished. And you can see that other one that's showing the anti-tumoral effect is way pronounced with the plasmid. We also checked in a different model, same outcome. So it was an anti-tumoral plasmid DNA. In this case, we targeted a xenograft of these MDA cells, which are a model for breast cancer. And they were expressing GFP. So what we did is we put a xenograft of these cells in zebrafish embryos, and we compared at 0 and 72 hours the growth or the well-being of this tumor mass. When we work with the scrambled or controlled plasmid, you can see that at 0 and 72 hours, the mass of cells was still there. When we used the anti-tumoral plasmid, reduced, and therefore there is this activity of the plasmid DNA. We also looked at mRNA, and we actually compared our formulations binding mRNA to the LNP composition. So we looked for the size and the polydispersity index, so how homogeneous these nanoemulsions were. They were very similar to LNPs. We also checked for the electrical charge. It was also on par with LNPs. And we looked at the stability in culture. Again, it was very similar. We looked at the association efficiencies with mRNAs. Of course, this is not quantitative, but with the agarogel, we could see again that the free plasmid was able to migrate when it was associated to both LNP and DIVERSA's lipid nanoemulsions. It couldn't migrate, so it was totally associated. And we even saw some efficiency of transfection. That's the graph on the right. In this case, we were using luciferase as a reporter, and we actually see that the transfection efficiency using lipid nanoemulsions of DIVERSA was a bit higher than with LNPs. But this is only one experiment, and we're repeating this. So what is our business model? We have 3 branches. The first one is we offer our technology as a ready-to-use reagent to the entire research community. So any research lab that doesn't have a pharmaceutical technology background or formulation background, they can get one of our kits. This is an example. And it's ready to use. You just need to resuspend the lipids, and then the protocol differs a little bit based on your type of molecule. Then we also do co-development projects, which are more geared towards biotech and pharma companies with a specific molecule. And we also do our own R&D. So basically, the kits are do it yourself. The co-development projects, do it with us. And the R&D is look at how we do. The reagents, they are 7 at the moment. We have one for cell tracking, which is the nanosystems labeled with a green fluorochrome, and they can be used for flow cytometry, fluorescent microscopy. And then we have kits for specific molecules, for small molecules, peptides, and proteins. All of them come in a version that's naked, and the other one has the green fluorochrome, depending on the type of experiments that you want. The reagents are sold through distributors. So at the moment, we're working with 3 scientific distributors, one for Europe, one for India, and one for Japan. And we are looking into expanding to other markets. Obviously, the US is the largest market for nanotechnology and drug delivery. So we would like to expand there in the near future. The co-development projects are basically more customized formulations for academic labs or companies, biotech, pharma, that have one or two candidate molecules. The way we do it is we do a feasibility study where we check that the molecule and our technology are compatible. So we get the molecule from the customer, we reformulate it in the lab in Santiago, and we send back the testable prototypes to the customer that then compares the original molecule with the formulated one. And the prototype that has the increased therapeutic activity, we can then enter a license agreement for a specific indication, because we can sub-license the use of our technology for specific indications. We are already working with a couple of companies, and we're open to collaborating with research labs and biotech and pharma companies. We also do our own R&D, especially in the areas of oncology, osteoarthritis, and metabolic diseases. We come from the nano-oncology department at IDIS, at the Health and Research Institute in Santiago. And osteoarthritis and metabolic diseases are also very good because our technology targets some of these tissues. We have key partners in different areas. For example, Asphalion for regulatory support, AMS Labs for manufacturing of the kits, Hoffman Eitel for intellectual property, and we have 2 CDMOs that can actually manufacture our nanosystems in GMP conditions, which will be important once these nanosystems have to go for a clinical trial batch production. The timeline, again, the company started in the first trimester of 2022 on January 17th last year, and we developed the reagents. We started the first co-development projects, and we're now looking into the territorial expansion to different continents. We've had lots of help for the internationalization from both EIT Health, which is EU funds for startups, and also ETHEX through the ETHEX Next program, which is a program from the Ministry of Economy and Commerce. You can visit us at our stand that we'll have during the 45th Congress of the will be here in Zaragoza. The latest, I'll be back in Zaragoza at the beginning of September. And we're actually, for the young or for everyone in the audience, we're hiring for different positions. So for research positions, we have a project leader and research scientist position open. And for my team, for the commercial team, we're looking for someone with science background, but also some commercial likes in Barcelona and Madrid. You can follow us on social media. We have the website, we're on LinkedIn and also on Twitter. And yeah, this is our contact. So, and you can reach out to me anytime. And any questions now, please. And thanks.
Thank you, Victor. That was a very interesting talk. I have lots of questions, but before that, I would like to give the opportunity to the people in the audience and also in the remote to ask questions. Okay, I'll start. Let's see how it goes. This is me. The first thing, there are several things that are interesting to me. That why your system seems to be better than, I don't know, a lipid nanoparticle. Why is it? The size is pretty standard. The composition, the outside composition is standard. So why it seems to enter better than lipid nanoparticles?
We have different advantages depending on what you compare us to. Compared to lipid nanoparticles, the main advantage is the easy composition. Like it's the less components means less regulatory hurdles. And also our business model is to share these nanomodulations. Lipid nanoparticles are patented by Pfizer and Moderna, and it's very difficult to get hold of them. Our business model is to share these through co-development projects and sub-licensing the technology. When it comes to the physical characteristics, for small hydrophobic molecules, the loading capacity is bigger than for liposomes, lipid nanoparticles, just because
the lipid nanoparticles also contain inside endosomal escape. And in your case, you don't need that. Apparently, you can get out of the exosome without any problem.
So
you get transfection there.
Yeah, we have not. It's a question that we get often. It's like, have you studied the mechanism of entry or how the cargo molecule is released? We don't have the detail. The main composition of the nanomodulations is sphingolipid, which is the lipidic monolayer, and then vitamin E, which is in the core. So there is a mix of fusion and endocytosis when they enter. Fusion because of the sphingolipid, right, which is prevalent in the cell membrane, and endocytosis because we've seen like the images in electron microscopy. How then these molecules escape the endosome? We haven't studied the mechanism.
But it's not built in. It's not built into the particles.
No, it just
happens.
Yeah.
That was good luck. And the biodistribution is also very interesting. Only 40% goes to the liver, which is
lower than
well, normally we get like 90%. So why is that? Why do you think?
It's a good question. I don't know. Actually, today I was talking to one of the researchers here at INMA, and he mentioned that, oh, you have a really good advantage because you do not use cholesterol in your formulations, which is important if you're treating some metabolic disease or I don't know why this distribution is lower in liver, but yeah, probably has to do with the composition.
Maybe that's also a possibility with a lack of electrical charge, because macrophages will detect that, protein will stick there. So that may also be I don't know.
Yeah. It's over my level, but I will ask and I can come back to you after the talk.
More questions? Yes.
Thank you. Very interesting talk. I would like to ask you if these particles you have used for a diagnosis as well. Like binding some tracer or something or protocol that can target in a specific tissues like cancer tissues or different. Yeah,
we actually have a publication on like using the nanomodulations as a carrier for a contrast agent. So yeah, it's one of the applications.
With staining, in vivo staining.
Yeah. I guess it's one of the applications that we haven't really commercialized yet. So we're focusing mostly on being the carrier for a therapeutic molecule. But as I mentioned at the beginning, we've been 1 year around, so we're very young and we're learning every day new applications. One of them is in cosmetic, for example, which it's like an industry that we didn't have in mind at all before we started, but we got lots of requests to carry different active principles, no, for cosmetic. So yeah, there's and contrast agent is another one of the applications.
More questions? I have a question. You only saw a few electron microscopy, but it seems to be very homogeneous. How do you regulate the particle size? Because everything is a batch system, so it's no microprocessing, no continuous processing, no micro mixing. So
no, it's basically the ratio between the sphingolipid and the vitamin E, but they are naturally very homogeneous. They will, if you have a specific ratio of the two components, they will all form in a very similar particle size.
So can you go higher and lower in particle size?
Yes.
Okay.
Yeah, we can. And it's important, for example, if you want to cross the blood-brain barrier or glomerular barrier in the kidneys, we can do them a bit smaller. If you want a cosmetic application and you don't want the nanomulsions to go past the epidermis, for example, we can make them a bit bigger to reduce the penetration.
The interesting thing that they don't seem to agglomerate, even though the electrical charge is small. Normally, you will expect them to agglomerate.
Yeah, yeah. Well, I didn't present it today. I can share more data, and it's all on our website. But we have studies with different small trucks and how increasing the truck load, other competitors would just start precipitating, and we are still able to accumulate these higher truck loads specifically for small trucks.
Of course, the big question is how do you target with these things? Because you try to link antibodies, peptides, things like that to the surface. Can it be done?
Yeah. So the surface is hydrophilic. So there we can bind targeting peptides or antibodies.
But in that case, you may attract the attention of the peptides. Because so far it seems to go mostly under the radar, but
no.
So how do you have any in vivo data for that?
No, we don't have. I was going to mention we haven't done like interleukin studies in vivo to see how the immune system reacts to our nanosystems in vivo. Something that's pending.
Very interesting system. Come on, more questions.
I have a question. You can listen to me. Yeah,
yes.
hello.
Yeah.
My name is Maria. Very nice presentation, very interesting topic. I'm wondering actually if you have any plans of developing a formulation to actually become like a dry product, like to freeze dry the particles later on, or this is a plan that will come like later on in the later development stages if it becomes a product.
So if I understood correctly, you're asking if we've tried it as a dried product for...
If you're planning to develop the formulation,
yeah,
to actually become a dry product, like to be able to dry the lyophilization process.
Yeah, so at the moment, the easiest application is a soluble administration or like an injectable. In the case of the reagents, the ready-to-use kits, we do supply them dried. So there is still one step where you need to resuspend them and add the therapeutic molecule. But it is possible to try them, and yeah, it's just a matter of combining it or making the process for adding the therapeutic molecule.
Thank you.
So a therapeutic molecule is loaded after synthesis, not during synthesis.
Yes.
Okay.
Yeah.
Okay.
Yeah.
I thought
It depends. So for a small molecule, you can add it to the buffer, and then while you're mixing with the ethanol and the buffer, it just gets encapsulated. For peptides and proteins and nucleic acid, it's like a second step after forming the adaptations because they're associated.
So what sort of loading can you get doing it after that? How much? By
Drug
weight?
load?
Yeah.
Weight. It depends on the molecule. So the small molecules that we've tried, up to 20%. Yeah, so between 5-20%, some of them less, but we can get to 20%.
Yeah.
Is my question a little bit related to what we were discussing now, and also about the I would wonder what are you doing in your R&D department only about the technology that you already have, try to doing do a screening about what kind of molecules you can start or bind with the technology. But or you're trying to or planning change functionalize them, changing that formulation to try to the encapsulation or encapsulate a lot of different molecules that has a lot of different properties without changing the properties of the technology. This is a really easy technology. Like let's say the best part. I was wondering changing the surface. Try to travel by change or the properties of the technology, or it's easy to modify it.
Yeah. So there is a limitation there. So whatever changes we do to the formulation, we need to play within the rules. Well, if we want the patent to protect that new development, it needs to be within certain rules. It has to have that much sphingolipid, that much vitamin E, and then you can add and take some things. But there's like a limitation of what you can do. In terms of what you can encapsulate, or you know, we have some projects where we encapsulate some metallic nanoparticles inside our lipid nanomulsions in case they're not soluble in aqueous solvents. We can also add antibodies or peptides on the surface. Sometimes we use surfactants to increase some properties like hyaluronic acid, or we use surfactants to protect the molecule that is hydrophilic to go inside rather than just stay associated on the surface. So yeah, I don't know if that answers your question.
Yeah. And also about the nucleic acid, siRNA. I didn't understand the percent. If you are studying
Yeah.
Because in studying.
No, it's just because we didn't. So from actually, like, the kits seem like it's just a kit, but there's a lot of work in getting a product that's going to be reproducible and working the same every time. So in terms of the mRNA kit, we're working. But for example, we can do the co-development projects. We have no problem in binding mRNAs or microRNAs. It's just that we don't have it in the form of a kit that you can use anywhere. But yeah, the kits at the moment, they're just for small molecule peptide. For us, it's a way that more people can try the technology, and also that they maybe as an entry step to the technology before doing a co-development project.
One last question related to the business side. So how do you see your company in 5 to 10 years time? I mean, if the plan is that a big pharma will buy you out and then you make a bunch of money.
I think this question is better for for the CEO, and
okay.
I'm just the first employee. So, but in terms of the commercial side,
or would you like a bit? I feel the question. Or would you like to you establish yourself as a provider of this technology to other people and not be bought out?
So I guess. So I'm not a co-founder.
Yeah.
If I were, it depends how much they offer. No? But it's a very exciting technology, and it just started. Like the patent was published in 2019, so we still have 15 years to develop the technology, and so it's at a very early stage. It's also that we have these two very different products. So from the commercial side of things, selling these reagents, working with distribution companies all over the world, and marketing these products is completely different to setting up collaborations with academic labs or with biotech and pharma companies and going into like clinical testing. So there's lots of different things going on. I think we'll see. Just the first year, we've learned a lot of things that work and the things that don't work, and we're just trying to get rid of the things that don't work. Thank you.
Any other question from the live or online audience? Yes.
Thank you for Ernesto. I'm curious about, you mentioned before that one of the molecules that you covered today are the peptides.
Mm-hmm.
How to purify the reaction once you couple the peptide, how you get rid of the excess peptide and the agents that you may use for the reaction, and how do you finally assess that the levels of these impurities are well threshold that they should be.
Yeah. So with peptides, as I mentioned, with proteins as well, we have two strategies for binding. One is a covalent bond, and the other one is electrostatic charges. In both cases, once the peptide is bound to the nanoemulsions, we can use microfilters. Like we use 10,000 kilodalton filters, the amicons, and basically you filter out anything that's smaller than 10 kilodaltons. That's what we recommend on using to filter out the unbound peptide.
And how do you check them for many
I don't know if I'm understanding the question, but usually for peptides, if we have like an analytical method, like an HPLC setup, we can check on the amount of peptide that's in one fraction or the other. Yeah.
Any other question? Yes.
I have a question. It's been a really nice talk. Thank you. When it comes to targeting, Have you ever tried any kind of stimuli responsive coating or binding with any molecule just to have a thermoresponsive nanoparticles or
Mhm.
something like that?
We haven't tried yet. It's interesting, and anything that we can associate to the surface or coat our nanomulsions with, we can try, but we still haven't tried the thermal or mechanical transduction.
No more questions?
Yeah.
If not, we thank Victor.
I
Yes,
don't know.
Yes.
Just last question. Hi, Victor. Thank you for this interesting lecture. I just first question. You mentioned that you need to produce.
Victor, you need to talk louder, please.
Oh, sorry. Okay, I'm close to my laptop, but.
Yeah, no, you're good.
Okay, so the question is, you need to produce your particles just using a mixture of ethanol and a buffer. So how easy just to remove the ethanol from the final formulation? Because I guess that's not so easy. It is required just to use ethanol, or you can just produce without the ethanol, just with a buffer?
That's a good question. I don't know. It's the method for forming the nanomulsions in this way is called ethanol injection, and basically the lipids are in the ethanol fraction first, and then by mixing with the aqueous solution, they will self-form, self-assemble. I don't think you need to remove the ethanol from the formulated fraction. It is at a very concentrated, like it's quite concentrated. So you will then dilute that in your final cell culture medium, or the medium that you use to inject animals, for example. But we do not ask for, there's no step for removing the ethanol.
Okay. Okay, thank you.
Yeah, it's a good question. I'll double check, but no, you can check our protocols online, and yeah.
Sure, I will check, because I was curious about that, because as Jesus mentioned, I was quite interested that you can produce, you can control the size of these particles without any other extra technology. So
yeah,
I thought that maybe because it was all the solvents, but solvents has some inconvenience like a toxicity, and so that
yeah, it's so in the protocol is it's a 10% of the total volume that you use in ethanol. So maybe that's why. So it's 100 microliters of ethanol, and then 900 microliters of your buffer.
Okay, great. Thank you.
You're welcome.
Okay. Okay, so we thank Victor again for his. We'll see you all in another Thursday spin-off talk. Thank you.
Thanks.